Good day, everyone. Today we are going to discuss about research methods in environmental science, particularly in environmental microbiology. The title of our research study is Ecosystem Restoration using Pongamia pinnata inoculated with plant growth promoting rhizobacteria or PGPR. I am Dr. Phoebe Nemezo-Calica of the Central Mindanao University or CMU in Bukidnon. Okay, so first let's describe Pongamia. Pongamia is a fast-growing, medium-sized evergreen tree. It has these light brown oval seeds, a cluster of seed pods, new pinkish red leaves, and pea-like blossoms. Now, the good thing about this tree is that it can yield up to 90 kilograms of seeds per tree, and each seed contains 40% oil, in which 50% is oleic acid, which is very suitable for biodiesel production. Now, aside from oil for biodiesel production, Pongamia pinnata can be used as medicine, flowers for honeybees, seed cake as green manure, soil reclamation, bark for paper and twines, feed for livestock, and for ornamental. Now, since Pongamia is a legume tree, it forms symbiosis with bacteria in the soil called rhizobia or rhizobacteria. Rhizobia belong to the family Rhizobiaceae, and they are genetically diverse. They nodulate plants under the family Leguminosae. They are generally chemoorganotrophs, meaning they utilize carbohydrates and amino compounds. They are also aerobic or microaerophilic. They have optimal growth conditions of 25 to 30 degrees Celsius and a pH of 6 to 7, and they are physiologically versatile. So our research question is, can Pongamia pinnata inoculated with plant growth promoting rhizobacteria or PGPR restore an ecosystem? So first, to answer our objective, to answer our research question rather, we first established the Pongamia rhizobia symbiosis. And in doing that, we first conducted soil analysis for the source of our rhizobacteria or rhizobia. Next, we have rhizobia isolation from soil and nodules. Next, we have characterization of the isolates. Fourth, strain symbiotic effectiveness test. And then of identification of selected strains. All of these were actually done prior to ecosystem restoration proper. Okay, so for soil analysis, we isolated 16 total of soil samples across Queensland, Australia, and we characterized them according to their physical properties, pH, nitrogen level, and also total nutrient analysis. After that, we isolated rhizobia from soil and nodules using baiting technique. So this is baiting technique. So we plant Pongamia in a sterile pots containing the soil samples from across Queensland, Australia. And after 8 to 12 weeks, we observed that there were nodules growing on the roots of the Pongamia plant. So all of these nodules were harvested, and then randomly we chose nodules to conduct this one drop one nodule technique, which is a technique to isolate the rhizobacteria or rhizobia or the potential PGPR isolates. Then we conducted substreaking to have a pure culture of the isolates. So we have a total of 95 potential rhizobia isolated, which all belong to the family Rhizobiaceae, based from the colony characteristics on the yeast extract mannitol agar. So yeast extract mannitol agar is the standard growth medium for the rhizobacteria. And we also observe that rhizobia nodulating Pongamia can be present in any soil at different sites in Queensland. And aside from that, based on their characteristics, so after isolating, we characterize them, and we found out that all of these rhizobacteria are actually aerobic chemoorganotrophs, and optimal growth is 28 degrees Celsius. They can grow at 0.1 to 0.5% sodium chloride medium and at a pH of 6 to 7. Out of the 95 isolates, 41 were fast growers while the rest were slow growers. Okay, so we characterize them as I've mentioned earlier, and we group them into fast growers and slow growers. So fast growers meaning they develop pronounced turbidity in yeast extract mannitol broth within 2 to 3 days, and their mean doubling time is 2 to 4 hours. The colonies on yeast extract mannitol agar were mucoidal and very slimy compared with the slow growers, and the diameter of the colonies of the fast growing strains were actually larger than the slow growing strains, which is 4 to 5 millimeters. And fast growers can be more efficient inoculant for Pongamia than the slow growers. They are more competent nodule occupants since they can outcompete the slow growers for infection sites on the Pongamia roots. And then they exhibited versatility in physiological characteristics, contributed to their adaptation to the competitive and complex soil environment, giving them advantage as effective inoculants for Pongamia. And lastly, legume trees such as Pongamia are mainly nodulated by fast-growing rhizobia than by slow-growing rhizobia. So slow growers, more than 3 to 5 days to produce moderate turbidity, and their mean doubling time is 6 to 7 hours. Colony size is from 1 millimeters. Now, the similarities between fast growers and slow growers were the following. They both utilize mannitol as carbon source. Their colony characteristics are the following. Shapes were usually discrete or round colonies, flat to high convex elevation, smooth margin, white opaque or milky to watery translucent, while some absorb the Congo red, making their colony color pinkish or reddish. So after characterization, we did the strain symbiotic effectiveness test to determine which among the 95 isolates was the perfect rhizobacteria or PGPR for our Pongamia. In a strain symbiotic effectiveness test, we found out that PRUQ05 produced the highest nodule number of plant, which is 143 nodules per plant. PR means Pongamia rhizobia, and UQ means University of Queensland, which is the source of our trees, and 05 is the number code. Okay, so this is the comparison among the uninoculated control, the positive control, and the PRUQ05. So it is very healthy, and the roots are very different compared with the positive and negative control. So aside from PRUQ05, which produced the highest nodule number, we also observed that PRUQ03 had the highest shoot length of 42 centimeters and had the highest shoot and root dry weights. So both PRUQ05 and PRUQ03 were chosen to be the isolates to be used in the ecosystem restoration. Okay, so before we conducted the ecosystem restoration, we identified first the two strains, the PRUQ03, PRUQ05, as well as the other two, top two slow growers, which are PRUQ01 and PRUQ04. So PRUQ01 and 4 were actually Bradyrhizobium elkani and Bradyrhizobium pachirizi or pachirizi. And then PRUQ03 and PRUQ05 were actually closely related to Rhizobium mesoamericanum strain. So in our ecosystem restoration, we use all of these strains, the top four strains, to inoculate in our Pongamia pinnata to be used in the ecosystem restoration. Okay, so this is the ecosystem restoration proper. So there were 3 stages of establishment of the plantation in a very difficult soil because it's a mining area. So this is in Miando Coal Mine near Kingaroy, Queensland, owned by Stanwell Corporation. And the planting was done by Bioenergy Australia and, of course, our laboratory personnel, yeah, including my supervisors, no, and me. We were there to plant all these trees. This was in 2013. So plantation tests were established to evaluate the suitability of Pongamia for rehabilitation, stabilization, and reforestation of coal mine spoils. Biofuel production is seen as an additional outcome. Remember, Pongamia can actually produce many seeds, and these seeds can be a source of biodiesel. So biofuel or biodiesel production is just an additional outcome of this ecosystem restoration. It's like hitting two birds in one stone. You can have a biodiesel and you can have bioremediation out of this Pongamia PGPR symbiosis. Okay, so note the sculptured soil preparation to increase water retention in the arid Queensland interior. So we have here elite trees with superior traits that are selected in Queensland as source of either heterogeneous seed populations or clonal material. Next, these are raised in nurseries to get established saplings, and then they are planted at approximately 450 to 600 saplings per hectare, and irrigation is needed if insufficient rainfall or extended drought periods are expected. Pre-harvest evaluation occurs in the first 3 to 4 years as some trees would already have fruited. Proper harvest occurs from the fourth year onward. First harvests are small and commercially insufficient but allow system development such as crushing and trans-esterification. And final products like oil itself and converted biodiesel are expected for commercial use by year 6. So here are the photos of our Pongamia inoculated with PGPR in 2018. So this was 5 years, now after 5 years. For our results and discussion, Pongamia pinnata has been found very effective in remediating not only overburdened dumps, but also the upper environment due to the tree's marvelous carbon sequestration capacity. Pongamia has been reported to be efficient in remediating problem soils containing heavy metals and other pollutants that impede plant growth. The fertility and productivity of the coal mine soil dump was restored using integrated biotechnological approach. Due to biofertilizer inoculation, different microbial groups, including the PGPR, which were practically absent in mine spoil, improved greatly, and the amendment and biofertilizer application provided better supportive material for anchorage and growth of the plant on coal mine soil dump. For our conclusion, Pongamia trees inoculated with PGPR have several benefits to restore the degraded land. First, carbon sequestration potential of around 49.28 tons per hectare, capable of withstanding drought stress, can grow on saline soils and needs little topsoil as it has a dense network of lateral roots and thick, long tap roots, can help alleviate compaction and crusting, a sturdy plant with no special nutritional requirements, can grow in extreme environmental conditions, tolerant to soil solidity, pH imbalances, high temperatures, heavy metal contamination, drought, and poorly drained soils. Phytostabilize iron, chromium, copper, manganese, and magnesium in fly ash dikes. Pongamia is being promoted as a soil retention and reforestation option for Queensland, Australia, as well as in the Northern Territory, Australia, due to its deep and broad root structure and their association with soil nitrogen fixers. And lastly, the choice of plants is a crucial aspect for the practical use of phytoremediation technique because not all species are capable of accumulating metals in their tissues, and most hyperaccumulators have small biomass and are slow growing. Thank you for your attention.